Hello, for our video today we are going to look at the lipids, and lipids are fats and oils that are present in living things. Now, just to put it into context, when we talk about the kind of nutrients that living things require, amongst other things, they require carbohydrates, proteins, and we often say fats, but from now on we're going to say lipids, which include fats and oils. In terms of carbohydrates, you'll remember that they contain carbon, hydrogen, and oxygen. Proteins contain carbon, hydrogen, oxygen, and nitrogen. All proteins contain these four elements. Often they contain other elements as well, depending on the kind of protein that you have. Lipids, similar to carbohydrates, contain carbon, hydrogen, and oxygen. The difference being, however, there are different properties for lipids compared to carbohydrates. So, for example, one of those is that they are insoluble in water. So lipids don't dissolve in water. They contain more energy for the same mass than carbohydrates. So if you took two similar masses of carbohydrate and lipids, you would find that the lipids contain just about double the amount of energy as compared to carbohydrates. And for that reason, that makes lipids ideal for storing energy that might be required at a later time. But that's not all they do. We have other roles for lipids as well. They act as an energy store, as we just said. They are also there for insulation. Some types of animal have more insulation than others. For example, whales will have a much thicker layer of fat below the skin for insulation compared to animals that live in a warmer environment. They're also there for protection. So for example, if you look at kidneys, they often have a thick layer of fat around them in order for protection. Plants have a waterproof layer over their leaves, which is lipid based, and also buoyancy. This means keeping afloat, and again this would be useful for animals that live in water, again going back to an idea of whales or maybe seals, something like that. Okay, so these lipids, fats and oils, they have various roles in living things. And what we're going to do now is look a bit more detail in terms of how they are structured or what the molecular structure looks like. So here I've got a little diagram to represent a droplet of a lipid, an oil in fact, and if we were to look in much more detail, in other words if we were to magnify many times, you'd see that the lipid is made of these molecules that look a bit like this. And this is something we looked at when we were doing our GCSE studies. We said that fats had a structure that looked like this, and if we magnify a bit more, we said that when we digest fats, they're broken down into these different parts, these three chains and this rectangular shape over here. And we didn't say much more than that. We did say actually that this part was called the fatty acid. So I've got three fatty acid chains here, and we said this part was glycerol. So when we digest our fats with our lipase enzymes, we broke them down into fatty acids and glycerol. Now what we need to do is to look at the structure of the fatty acid chain and the glycerol in a bit more detail and look and see how they actually combine to produce our lipids. Okay, so let's have a look a bit more detail at the fatty acid part. The fatty acid part of the molecule contains this hydrocarbon chain, which I've drawn over this end, but on the end we have what's called a carboxyl group. A carboxyl group, COOH. Now in terms of the fatty acid chain, I've just got a dotted line here. I've been a bit lazy and not drawn out all the carbon atoms, but typically you'd see maybe 15, 16, 17 carbons in the fatty acid chain that are present in our lipids. There are other types of lipid with more, maybe less, but typically 15 to 17 that are found in living things. Okay, so that's what the fatty acid part of the molecule looks like. The glycerol part looks a bit more like this. We've got three carbon atoms, each with an OH group attached, and hydrogen atoms on the other side. So this is our rectangular part of the molecule that we call glycerol. The structure is actually like this, and our fatty acid chain or fatty acid part looks more like this. Now in terms of how they combine, the fatty acid chain is often drawn with the carboxyl group like this, COOH, rather than the structural version that we've got over here. And the reason why I've drawn it like this is it just makes it easier to show what's going on when the fatty acid combines with the glycerol. Now this is very similar to what happens with proteins and carbohydrates that you saw in previous videos, but what we have is a reaction that combines these two together, and it's called a condensation reaction. And just like with protein and carbohydrate, the condensation reaction involves removing water. So what we have is a reaction that removes a molecule of water. Now I must get this right. H2O. So that part there is removed, a molecule of water is removed, and we have a condensation reaction, and that will actually produce what's called an ester link or an ester bond. You can use either one of those terms, but the key part is it's an ester link or an ester bond. And what very often happens is you get 3 of these fatty acid chains joining with the glycerol, and we end up with something called a triglyceride. And in terms of the triglyceride, we have 3 fatty acid chains. Sometimes they can be different chains, sometimes they're similar chains, but the point is if we have 3 of these fatty acid chains joined onto our glycerol, we have produced a triglyceride through our condensation reaction. Importantly, when we're breaking it down as we did over here, the opposite of a condensation reaction is hydrolysis. Hydrolysis. And that is when we actually add water, a water molecule, and actually break this fatty acid chain away from the glycerol. Okay, so condensation reactions produce these links, these ester bonds, and hydrolysis can actually break them in order to break it down into the different parts. Now, when we actually have this molecule, it looks a little something like this. Now, this all looks very complicated. I've got my 3 fatty acid chains here, 1, 2, and 3. In this case, they're all the same, but if they join through our condensation reactions to our glycerol, what you'll end up with is something like this. Now if you look on this part here, you can see how it's changed. 1 molecule of water has been removed, and the bond now looks like this. The ester bond looks like that. So while it looks quite complicated, the majority of the molecule you don't really need to know and memorize how to draw. The important part is being able to show a condensation reaction happening between the OH group and the carboxyl group of the fatty acid. And because that's the important part, you may see the structure of the fatty acid drawn in various ways. So for example, you might see it drawn like this, the carboxyl group on the end there being very important, and the hydrocarbon chain just drawn as an R to show that it's a group attached to that. And if you saw that, you would do the reaction in the same way to show how the bond occurs. Sometimes we see the glycerol shown like this. It's a different way of representing the same thing, but if we were to show the reaction there, it's very similar. It's the H, in actual fact that way around, H2O removed like so, and you can just go ahead and show it in a very similar way to what we've just done. The next thing I want to look at is this idea of saturated and unsaturated fatty acids, these fatty acids that are present in the lipid molecules. And here I have an example of a fatty acid that is saturated. And by saturated, we mean that there are no double bonds between the carbon atoms. So a saturated fatty acid has no double bonds between the carbon atoms. But when we look at a saturated fatty acid, you will see 1 or more double bonds between the carbon atoms, and they would look like this, simply drawn as 2 lines between 2 of the carbon atoms. One of the hydrogens is gone because that bond has been taken up between the carbon atoms there. Now the effect that this has on the molecule is that it changes its shape slightly. What it does is it actually produces a slightly more pronounced bend in the fatty acid chain. Normally we draw the fatty acid chain as a crinkle or pleated kind of shape, but what this does when we have a double bond is it makes it have a much more pronounced bend in the molecule, so maybe like this. And this is important in terms of determining whether it's liquid or solid at room temperature, which I'll talk about briefly in a moment. But here we have our saturated fatty acid. Because we have only 1 double bond between carbon atoms, we can say that it's monounsaturated, mono meaning 1, and if we have more than 1 double bond between carbon atoms, we can say it's polyunsaturated. So the key thing here is if we're talking about saturated fatty acid chains, we mean that they have double bonds between the carbon atoms. I have seen a question that on a past paper that asked what these unsaturated fatty acids were, and it was 2 marks. One was for mentioning the double bond, and 2 for saying it was between carbon atoms. If you're going to be more specific, monounsaturated and polyunsaturated. Mono means one and poly means many. We did mention about the bending in the fatty acid chains, and if we have a lipid that has a lot of these double bonds causing a lot of bends, we might see a triglyceride that maybe has bends like this. And what that means is, because of the bend, the molecules physically can't get as close together as if they were saturated, and this means that the lipid is more likely to be liquid at room temperature. Triglycerides that have saturated fatty acid chains can actually squeeze up closer together, and therefore they're more likely to be solid at room temperature, and we tend to call those fats as opposed to oils. So some key terms there that you need to be able to describe and explain what they mean. The last thing I want to do very quickly is quite simple. You don't need to spend a lot of time on this. That's the test for lipids. How do we test if a substance contains lipid or not? Well, we take a test tube with our sample, could be this over here. We add some ethanol, we shake very vigorously, we add some water, and if we get a result that shows a cloudy white suspension, so that might be like this, that indicates the presence of a lipid. If it stays clear, then we would think that there was no lipid present. The cloudy white suspension is a result of an emulsion being formed between the water and the lipid. Okay, so that's the test. I don't need to spend a long time on this. It's just a question of learning it, so I'll leave that there with you. Other than that, that's me done for the video today. Thanks for watching, and see you soon.